Hey bio students, hey, we are going to be doing some dihybrid crosses, okay? And for those of you that are caught up and you're staying up with the lessons, you're going to do well. For those of you that have not been doing your monohybrid crosses, you haven't looked at them, you haven't done the worksheets, you haven't checked them, you're going to struggle, all right? So be on top of your assignments. When we return, I'm going to collect all of that stuff, okay? All right? So, hey, before I go too much farther, I want you to be able to finish your lab reports. So let me give you some dominant traits on your lab report. So if you want to pause the video and go get your lab sheet, and I'll give you your results. All right, so here are the actual frequencies or the dominant recessive traits that you guys did on your lab report, and then you can finish writing up. I would go ahead and just, if you got it right, great. If you didn't, mark it. You're not going to lose points because you guessed wrong, all right? We'll explain in a minute. All right, so tongue rolling's dominant. I'm just going to go straight down the list. Widow's peak is dominant. Knuckle hair's dominant, earlobes dominant, straight thumbs are dominant, bent pinkies are dominant, left over right dominant, chin cleft dominant, short big toe is dominant, separate eyebrows dominant, and freckles are dominant. Now, you probably did not get those all right, and so that's why you want to answer your questions on number 2. Did your results differ from what was expected to be dominant or recessive, and why? All right, so why did you get them different? There's some reasons for that. I mean, obviously, some of these traits can just be rare in our population. And the other thing is maybe you guys read them wrong. It's a pretty subjective test, so maybe you just read them all wrong. All right, question number 3 asks, if you did this experiment with the whole school, would you expect them to be more or less accurate? That's kind of a toss-up. I mean, you're still testing the same population. You could still get the same results, or the more that we test, the more accurate the probability we would expect, right? So a couple ways you could answer that. So did you have any genetic traits like anybody else in class? You're supposed to go around and check. You may not have gotten time to do that, all right? So now you can kind of finish that lab report, and then when we return, I'll collect that, okay? All right, so that's your human genetics lab. We will now go on to dihybrid crosses. There's a copy of these notes for you to go through as we are doing this in class. So, all right, so let me change this camera for you now. And, all right, I got this stuff out of the way. Let me get this down. I will rotate it so we can do some notes. All right. You should have a copy of these for you to look at. All right. So I'm just going to kind of go through what a dihybrid cross is. A dihybrid cross is going to have 2 traits. We have been doing monohybrid crosses, and that's just 1. But now we're doing dihybrid. All right, 2 traits. And so what I did is I picked one of the traits from the Mendel's pea plants, just so it's something that's familiar that we've worked with. I will pick some other ones here in just a bit. All right, so for example, we're just going to do a homozygous dominant green and tall. Homozygous dominant green and tall, so it'd be 2 big G's, 2 big T's. We're going to cross that with a homozygous recessive yellow and short, so that's homozygous recessive yellow and short. For those of you that have done your monohybrids, you understand what I'm saying here. So now what we want to do is we want to split these up. We want to put one of them here and one of them here. But we want this to go in a particular order. And so what I want to do is I want to kind of show you what that order looks like. All right? So here's what this order looks like. All right, this is what's called the FOIL method. All right? Where that means first, outer, inner, and last. All right? First, outer, inner, and last. And so the way that's going to line up is when we actually foil this, we're going to put the first here, outer, inner, and last, just so we kind of get a method to it. And then when I come over here, this is going to be first. This is, so you see first G, first T, and then this is going to be outer. So that would be the outer G, outer T. All right. This is going to be inner, so that would be the this one. Inside G, inside T, and then this would be last, so it would be this line right here. Last G, last T. Outer would be this G, this T, first. All right. So when we put that up here, it's going to look, you know, something like big G, big T, big G, big T. So I'll go back to here, and if I follow that method, then I'm going to end up with big G, big T, big G, big T, big G, big T, right? And so I would do that foil. I would do that foil for this one as well, right? First, outer, inner, last. Got it? First, outer, inner, last. And so those would be like this. Now what we want to do is we want to push all the G's and T's down and push all these G's and T's across, and you want to put them back together. And you want to put the dominant, if it looks like this, you want to put that. We don't want it like this. We don't want it to look like that. All right, we want to put the traits back together. We also don't want it to look like this. All right, it's pretty common that you want to write the dominant traits first. So neither one of those we want it to look like this. And actually, they're all going to look like that. And so what I have is, I've actually got one that I've got all filled out here. So all these, and you can see they're all filled out. And now what we want to do is we want to read off the the genos and the phenos. So when I look at these, I usually make a check mark. That's heterozygous for both traits. So I would write heterozygous for both trait. Go ahead and write big G, big T, little t. And you can see that 1, 2, 3, 4. All of these look exactly the same. You're going to have a couple of these on your packet, so that'd be 16 out of 16, 100%, if we wanted to go a percentage. And then I would look at the phenotype, green and tall, so they would all be green and tall. And then I usually come through and I put a line through here just to make sure I've checked them all. So all of them, 16 out of 16, are green and tall. So that's kind of basically how you do a dihybrid. Just real quickly, just kind of showing you how we talk about it and how we foil it and things like that. So, all right. So what I want to do now is I want to kind of maybe move off of that, and let's do an example of say one out of your notes. Let me run through the ugliness example. All right, ugliness example. And so what I'm going to do is I'm just going to kind of do this example that's in your notes. All right. So, and I'm just going to kind of just reveal it a piece at a time. All right, so this would be kind of a problem that you might get, say, on a quiz or a test or something like that. So it says ugliness, big U, is a dominant trait in rural communities. A trait that often shows up with ugliness is the dominant kindness gene, which is a big K. What is the chance of getting, let's go ahead and underline this, an ugly child with a nice personality from a mom who is homozygous, ugly, and heterozygous, nice, and a dad who is beautiful and mean? So the first thing we got to do is we got to be able to get the parents. Mom is homozygous, dominant, ugly, right? So she would be 2 big U's. And it says that she is heterozygous, nice. So she would be a big K and a little k, right? And then a dad who is beautiful. Well, if ugliness is dominant, then dad has to be 2 little U's. and he's mean, and kindness is dominant, so he has to be 2 little k's. Yeah? So I made them real small just so you can see the difference. So what we would have is we'd have some parents that would look like this, right? Yeah, and then we'd want to foil these. So if I was to foil these, then I would end up with, I'd have U, U, K, K, and I would have, let me go ahead and do a different color here. All right, that would be the first. All right, and then we would have outer, and then we would have inner, and then we would have last, right? All right. All right, back to it. I had a phone call I had to answer. All right, so back to it. So we would look like this, and then we would want to foil these like this. All right, so one parent would go across the top. You can see this is the first, big U, big K right here. And then we have outer, big U, little k. And then we have inner, big U, big K. And then we have last, big U, little k, big U, little k. See that? And then when we foil this one, that one's going to be easy because they're all going to be the same. Little u, little k. First, outer, inner, last. All right? So that's how that one's going to look. All right? And then when we fill this thing out, all right, this is how it's going to look filled out. So we put the big U's in, we put the little U's in, we put the big U's, and we put little U's and little K's, and we fill this thing out, and you got to make sure that you don't mess up here. And so now it's time to read off genos. That's heterozygous, and we've got 4 that look heterozygous. We got heterozygous, homozygous, recessive. We got 4 that look like that. We got heterozygous, another 4, so that's 8. And then we got heterozygous and homozygous recessive. That's another 4. So we would end up with hetero for both to be about 50% of the time. That would be 8 out of 16, 50%. And then we would end up with hetero and homozygous recessive, which would be these ones. That would end up to be 8 out of 16. All right? Okay, now we want to read this off. Ugliness is dominant. So this one right here, we have ugly and kind. Ugly, kind, right? Not this one, ugly, kind, ugly, kind, ugly, kind. So that is also going to be 50%, right? 50% ugly and kind. And then we've got 50% for the other ones. We got ugly and nice. They're ugly, but they got a nice personality. And so that would be what we get here. We got ugly, nice, 50%, ugly, mean, 50%. So then the question asks, what's the chance of getting a child with a nice personality, an ugly child with a nice personality? Well, my circle's a little bit big here, but we would want to circle that one right there. That's the one we want to go with. All right? So, dihybrids are pretty tough. So take your time, all right? Take your time. I left you a worksheet that's got a few on there for you to work out, okay? I'm gonna post the answers to that worksheet probably tomorrow so that you can kind of understand what we're doing on dihybrid, and I'll probably work out a couple of those, okay? All right? All right, and you have some dihybrid problems that show up in your worksheet, so maybe that'll help you a little bit on that. All right, you guys? All right. Hey, take care.